Hello, dear students. Welcome to the e-learning platform of Sanjivani Mahavidyalaya Chapoli. Here is Department of Botany for BSc second year and BSc third year student. Semester 4th and semester 6th. Paper 9th and paper 14. Plant metabolism, biochemistry and biotechnology. In previous lecture, we discussed the evidence in support of two phases of photosynthesis, that is evidence physical separation of chloroplast into grana and stroma fraction, temperature coefficient, evidence from intermittent light, evidence from carbon dioxide that in dark. And now in this lecture we are going to discuss some light reaction or photochemical phase of photosynthesis. The light reaction or photochemical reaction takes place in thylakoid membrane or granum, and that reaction is completely dependent on the light, and that's why it is known as light reaction or photochemical phase. That photochemical phase or light reaction is carried out in the thylakoid membrane or granum of the chloroplast, and it is completely dependent on the light. The raw material for these reactions are pigments, water and sunlight. The reaction of photosynthesis is carried out in the chloroplast or chlorophyll. That chlorophyll is the pigment. So it is one of the raw material for the photochemical reaction. Apart from the pigments, the water and sunlight is essential. That is, the pigments, water and sunlight are the raw material for the photochemical reaction or light reaction. That photochemical reaction can be summarized in three steps. First, excitation of chlorophyll molecule. Then photolysis of water is carried out. And after the photolysis of water, photophosphorylation is carried out. Let's see one by one. First, excitation of chlorophyll molecule. Of chlorophyll molecule is the first step in the light reaction. The special type of chlorophyll, that is P680 and P700, the two pigments that receives quantum of light. The reaction center 680 and 700, the two pigments that receives the quantum of light, and then it becomes excited and release electrons. Second step in the light reaction is photolysis of water and oxygen evolution. That is the Hill reaction. Before 1930, it was thought that oxygen released during photosynthesis come from carbon dioxide. Before 1930, the oxygen released during photosynthesis is come from carbon dioxide. But for the first time, Van Neel discovered that source of oxygen evolution is not carbon dioxide, but it is water or H2O. In his experiment, the Van Neel used green sulfur bacteria which do not release oxygen during photosynthesis. They release, instead of oxygen, the sulfur is released. These bacteria require H2S in place of H2O. The oxygen evolved during photosynthesis, that is, come from the water molecule and not from the carbon dioxide molecule, and for that purpose the Van Neel carried out the experiment. He used green sulfur bacteria, that is the photosynthetic bacteria. The photosynthetic bacteria, the green sulfur bacteria, use sulfur for their energy. So during the photosynthesis, he evolved the sulfur molecule. The idea of Van Neel was supported by Robert Hill. The Hill observed that the chloroplast extracted from the leaves of Stellaria medium and Laminium album, when suspended in the test tube containing suitable electron acceptor, that is potassium peroxylate or potassium ferricyanide, oxygen evolution takes place due to photochemical splitting of water. The splitting of water during photosynthesis is called photolysis of water. The splitting of water molecules in the photosynthesis is called photolysis of water. Manganese, calcium, and chlorine ions plays the important role in the photolysis of water. This reaction is also known as Hill reaction. To release one molecule of oxygen, two molecules of water are required. The evolution of oxygen from water was also confirmed by Rubin, Randall, Hassid, and Kamin in 1941 by using heavy isotope oxygen 18 in green alga chlorella. The splitting of water molecules during photosynthesis is called photolysis of water. Robert Hill carried out some experiment. He supplied suitable electron acceptors instead of carbon dioxide or in the place of carbon dioxide, and he found that oxygen is still evolved, and he confirmed that the oxygen is evolved because of the splitting of water molecules. From one molecule of oxygen, two water molecules are required, and that findings of Robert Hill confirmed by other scientists also, that is Randall, Rubin, and Kamen in 1941 by using heavy isotope oxygen 18 or labeled oxygen, and that carried out the experiment in green alga chlorella. That reaction is known as Hill reaction. When the photosynthesis is allowed to proceed with hydrogen, and that hydrogen oxygen is labeled oxygen is used, that is 18 O18, and normal carbon dioxide is used. The evolved oxygen contains heavy isotope. If photosynthesis is allowed to proceed in presence of carbon dioxide 18 instead of the labeled oxygen is incorporated in the carbon dioxide and the normal water is used, the heavy oxygen is not evolved, and he confirmed that experiment confirmed that oxygen is come from the hydrogen and not from the carbon dioxide. Next step in the photosynthesis of is Photophosphorylation. Synthesis of ATP from ADP, adenosine triphosphate and adenosine diphosphate. Synthesis of ATP from ADP and inorganic phosphate in presence of light is known as photophosphorylation. It is clear? The synthesis of ATP from ADP in presence of inorganic phosphate and light, that is known as photophosphorylation, and it was discovered by Aradwad and his co-workers in 1954. Photophosphorylation is of two types: cyclic photophosphorylation and non-cyclic photophosphorylation. We discuss the first, cyclic photophosphorylation. It is a photophosphorylation in which the electron expelled by the excited chlorophyll molecule or photosystem's center is returned to it after passing through a series of electron carriers. What happened during cyclic photophosphorylation? When light trapped by the chlorophyll molecules, the reaction centers, that is photosystems, are excited and the electrons expelled from the photosystem or photocenter, that is photosystem first, is carried out, or here is photosystem 1 is activated. The electron expelled from the photosystem 1 is passing through a series of electron carriers, and finally the electrons comes to the original place, that is the cyclic photophosphorylation. That cyclic photophosphorylation or electron transfer from series of electron acceptors, it occurs under conditions of low light intensity, the wavelength longer than 680 nanometer, and when carbon dioxide fixation is inhibited. When that cyclic photophosphorylation is carried out, that condition, that low light intensity and carbon dioxide fixation is inhibited. Absence of carbon dioxide fixation result in non-requirement of electrons, as NADPH2 is not being oxidized to NADP. Cyclic photophosphorylation is performed by photosystem 1 only. Its photocenter, that is P700, extrudes an electron with a gain of 23 kilocalorie per mole of energy after absorbing a photon of light. Cyclic photophosphorylation is performed in photosystem 1 only, and the reaction center of that photosystem 1 is P700. That P700 extrudes an electrons with a gain of 23 kilocalorie per mole of energy after absorbing a photon of light. After losing the electrons, what happened? When he absorbed, the P700 absorbs the light, the photosystem 1 is excited and the electrons is expelled from that. The expelled electron passes through a series of carriers. that expelled electrons is passes through a series of carriers including special chlorophyll molecules FES, ferridoxin, plastoquinone, cytochrome BF complex, and photocyanin before returning to the photocenter, which is the electrons carriers. That is the special type of chlorophyll, that is chlorophyll or X, FES, ferridoxin, plastoquinone, cytochrome BF complex, and plastocyanin are the electron carriers. From that carriers, the electrons return to its original photocenter. What happened when the electrons carries from one carrier to other carrier? The electron loses sufficient energy from carrier to carrier, and by losing energy the ATP is formed from ADP and inorganic phosphate. By using ADP and inorganic phosphate, ATP is formed. Halobacteria or halophile bacteria also perform photophosphorylation, but ATP thus produced is not used in synthesis of food. This bacteria possesses purple pigment. That bacteria contains the purple pigment attached to the plasma membrane. As light falls on the pigment, it creates a photon pump which is used in ATP synthesis. And here we stop and we discuss on the non-cyclic photophosphorylation or Z scheme in the next period. Thank you.